Good morning, students. This is Miss Tittle, and today we are working on Inquiry 3.1. We're going to be drawing and measuring a blackworm. So for this assignment, you're supposed to watch this video and then complete your lab report. So let's take a look at your lab report. You just open it with Cami, and on your lab report, it says that our question is, what are the characteristics of a California blackworm? Okay, so think about it. We know from its name that it's a worm, so that's a very big clue. So for your hypothesis, you would say if a California blackworm is a blank, then it will have blank. Well, you could put several different options here in this blank. You could say it's a worm. You could say it was an invertebrate animal. You could probably come up with some other ideas there too, but either invertebrate animal or worm or something like that would be an appropriate option for this blank. So if it is a worm or if it is an invertebrate animal, then what would be something that it would have that would be characteristic of that type of organism? So figure out something that you would like to put here that would be a clue as to why it would be a worm or why it would be an invertebrate animal or what other option that you chose. Okay, let's look on down on our lab report. Our materials on this lab are found on page 41 in the textbook, and our procedures are found on page 42 to 44. So let's take a look at our procedures and see what we're going to be doing. Okay, in the book I have turned to page 44, which is page 59 on the blue slider, and the procedures for this lab say to use your pipette to dampen filter paper with a few drops of water from the culture container, and to place the petri dish with the blackworm on it on the microscope stage and focus it on 40x. And we'll take a look at that in just a minute. And what you're going to be doing is preparing a detailed drawing of your blackworm. You have the template to where you should draw your blackworm on your lab report. You'll be drawing what you see through the microscope lens right here in this circle. And then our procedures say that you will be labeling your drawing. The title of your drawing would be Lumbriculus, the California blackworm. So that would go on the second line. Remember, we're looking at it on 40x, so that's what would go on the first line. So let's glance back at our lab report for a second. Here you would draw what you see through the microscope lens. On this line you would put what power of magnification. In this case we're using 40x. And then on the bottom line you would put the name of what you're looking at. Remember to write it in all capital letters according to our guidelines for scientific drawings. Okay, after we do that, the next step says for you to label at least 3 structures of the blackworm on your drawing, and you can find those structures on page 49 to 53 in the book. Actually, page 49 would be where it shows the picture. Right here, this picture shows the parts, but I have given you a little bit of a shortcut. If you'll look back out to the assignment and scroll down, I have given you a shortcut to that picture right here. So you have your microscope view of the blackworm on 40x, and you also have the diagram that you would use to label your microscope drawing. Okay, let's go back to our procedures. Okay, so label at least 3 structures of the blackworm on your drawing. And then the next thing it asked us to do is to measure our blackworm. So I'm going to switch over to the webcam for just a moment, and we will take a look at the blackworms in person. So I'm going to switch over to the webcam. As you can see in this bowl, we have the California blackworms, and they're all over here in a big group. They do that for several reasons. They like to group together for safety, but also they like to group together for probably warmth, because the water temperature is a little cool right now because it's cold weather, winter time. So they're all kind of bundled up together. Now over here you can see one black worm that's kind of swimming around by itself. You can see how it's stretched out. They're very long considering their body size, but they are really small organisms. See, right here we have this pipette, which y'all know is a small object, and in comparison the black worms are tiny, but then when you look at them here in the water and they're stretched out, you can see how long they are in comparison to how small and microscopic they are. Okay, I also have one of the black worms on this petri dish, but he's coiled up tight right there to protect himself. So I'm going to get a little water from the culture container and I'm going to squirt it on him and try to straighten his body out. As you can see, he doesn't want to be bothered, but we're going to try to straighten him out straight enough that I can get an estimate of how long he is. Hang on, let me focus my... There we go, we got our webcam focus there. Okay, I'm trying to use the centimeter side of my ruler, and this black worm is about 3 centimeters, which would be 30 millimeters. Okay, so our measurement for our black worm is about 3 centimeters. Okay, as you can see, a few of them are crawling around. Now here's something that usually gets some excitement from the class. I'm going to disturb them a little bit with the petri dish and let you see them swimming around. You can see all the little black worms swimming around. We just disturbed the water a little bit and it stirred them up. They're all going to be just wiggling around, swimming around until they group back up again in a little clump. They like to do that. I wish y'all were here in the classroom to see them in person. They should still be alive by the time y'all get to come back to school, so you'll get to see them then. I'll make sure we get them out and let you look at them. Okay, let's go back to our lab report. Okay, on our lab report, after you've done your microscope drawing, then you would need to scroll down and answer the analysis questions. Why do the black worms make some of their unusual movements? And black worms have a more complex way of moving food and oxygen through their bodies than most microorganisms. What evidence have you seen to support this? Well, let's take a look back at our microscope drawing here. You can see through their body. Their bodies are translucent, which means you can see through them. So what we're seeing through their skin, we can actually see this blood vessel running through, and we can actually see these segments of their body. They are a segmented worm. So we can see the segments, their digestive tube, their blood vessel. Those things are pretty obvious to us. And as we see on the diagram, it has the blood vessel and digestive tract labeled there. Now, they do have little hairs on their body called KT. They're kind of like little whiskers, but they are so tiny that you really can't see them from even the microscope view. But their bodies have those little bitty hair-like structures called KT on them. Okay, they have an anterior end and a posterior end. And we usually can pick out the anterior end because if they're moving, that's going to be the end that heads first. They're going to move forward with their head end. So that's usually the way you can tell is from their movements. You can see that their head end goes forward first. Okay, let's look back at our lab report. After you've answered your analysis questions, you have some vocabulary words. Your first word, Lumbriculus variegatus, is the scientific name of the blackworm. Lumbriculus is the genus name, variegatus is the species name. So Lumbriculus variegatus, you see it's written in italics because it's a scientific name. And scientific names are a two-part name that include the genus and species. So this particular species is called Lumbriculus variegatus. Like we mentioned, they are a segmented worm. They are aquatic. They live in water. The KT are their little hairs. The blood vessel transports blood and nutrients through their body. The anterior end would be the head end. Posterior would be the tail end. And then their digestive tract is what moves their food through their body. Now, what the blackworms most likely eat are little micro organisms and microparticles that would be in the pond where they live. They generally live in freshwater ponds and creeks. They'll be usually along the banks of the creek or river or pond. And they're so tiny you probably have not noticed them before. Okay, and then lastly on your lab report is for you to write your conclusion. Your conclusion should be a paragraph, at least 5 sentences long. It should restate the question of the lab, which is what type of organism or what are the characteristics of a California blackworm. Then you should tell me if your hypothesis was correct or not, and then go into a little detail about what the blackworm is, what its characteristics are, and you can base your conclusion on the evidence you gathered from the diagram we looked at, your microscope drawing, and then the live footage of the blackworms that you saw. Okay, if you have any questions about this lab or lab report, then make sure you message me. I hope you're all doing well and are healthy. I'll talk to you later. Bye-bye.